Hi children, we are into the 13th chapter, photosynthesis in higher plants, and we are starting off with early experiments towards photosynthesis. We have some very pioneer experiments, early experiments, first conducted by Joseph Priestley. What did he prove? The experiment itself is very clear from the diagram. I will just zoom through the diagram displayed. In his experiment, he put a burning candle, a live rat in a bell jar that was sealed with Vaseline so that it was airtight, and you can expect what to happen. After some time, the candle got exhausted and the rat got suffocated and died. So he knew something. It was way back in 1774 when oxygen or carbon dioxide was not at all identified as a gas that is evolved or given out during respiration, or the gas that is taken during respiration. Okay, so he understood from this very first step, he understood, or this experiment, he understood that the breathing animal, rat, and the burning candle polluted the environment. We know how did they pollute. They gave out a lot of carbon dioxide, and when a lot of carbon dioxide was present in the bell jar, it got exhausted. Carbon dioxide was filled because oxygen was utilized for burning and oxygen was utilized for breathing by the animal, and finally the animal died due to suffocation and the candle got exhausted because there was no oxygen for burning. We know for combustion oxygen is needed. So from this he understood something polluted the environment due to the breathing of the animal and due to the burning of the candle. In the second part of the experiment, he included one more specimen, a mint plant. A healthy potted mint plant was introduced along with the animal, rat, and the burning candle. And he found, after some time, he found all of them hale and healthy. The animal was alive and agile, the burning candle was continuously burning, and the plant also was intact without any problem. So from this he understood something, and he hypothesized that plants restored to the air whatever is removed by burning of the candle and by the breathing of the animal. Clear? So he understood that the plants restored, we know what was restored, oxygen was restored. He restored something that was removed by the burning candle. We know for burning of candle oxygen was utilized, and as well as for breathing also oxygen was utilized. That was restored by plants. We know that plants give out oxygen, and animals and the burning of candle utilizes the oxygen and give out carbon dioxide. So he was the first to identify that the plants is restoring something that is removed by the burning candle and the breathing of an animal. And later he himself discovered that what was evolved during photosynthesis is a gas called oxygen. Later, in his later experiments, this was a very early experiment of his. The very same experiment was conducted by Ingenhousz. Next scientist, Jan Ingenhousz. He conducted the very same experiment, but only thing, one he conducted under sunlight and the other in dark room. And he found out that along with this, light is necessary for photosynthesis. See, if I am zooming this, the very same setup. Under the bell jar, there was a burning candle and a potted plant. It was left in the sunlight for 7 days, and the candle continued to burn and the plant was alive. But the very same setup he put in the dark room. He did not expose it to light, he put it in a dark room, and he found that after 7 days the candle got exhausted and the plants got wilted. So he understood that sunlight is very essential for photosynthesis. Next experiment was by T.W. Engelmann. T.W. Engelmann, using a prism, he split white light into its 7 spectral components. We know the 7 colors: Vibgyor, violet, indigo, blue, green, yellow, orange, and red. and this was made to illuminate a suspension of an algae called cladophora along with a bacteria, aerobic bacteria. So, once again using a prism, he split white light into its 7 colors. You all are very well aware when white light is passing through the prism, it gets split into its 7 colors. So, I am zooming through this picture. As I zoom, the right up becomes blurred. Bear up with that. So, the prism is used, see the prism I am highlighting is used to split light. So, there is a source of light here, okay, maybe an artificial source of light. It was used to split white light. We know white light very clearly, it should be white light. It was split into its 7 colors, and you can see the split of the 7 colors starting violet, indigo, blue, green, yellow, orange and red. And this was made to illuminate a suspension. So, I am just enhancing the diagram. This was made to illuminate a suspension of an algae, which I am going to represent with green dots. So, algae, the name of this algae is cladophora, and along with this cladophora there were the aerobic bacteria, which is going to be indicated in, say, black color. They are found equally distributed, the aerobic bacteria. Okay, so I put those black spots equally distributed in the entire container. Okay, so I am just marking the parts. The green stands for cladophora. It is an algae, and the black dot stands for the aerobic bacteria. So, you can understand why aerobic bacteria is used. It could have been anaerobic bacteria. So, here the aerobic bacteria acted as an indicator. It served the purpose of an indicator of oxygen evolution, to detect wherever oxygen is present. So, once again the experimental setup, a prism was used to split light into its 7 colors, 7 spectral colors, and this was made to illuminate a suspension of cladophora with aerobic bacteria. Clear? So, did you understand the experimental setup? Once again I am reiterating, with the help of a prism, white light was split into its 7 spectral colors, VIBGYOR, and this was made to illuminate a suspension of cladophora and aerobic bacteria. The purpose of aerobic bacteria was to identify or detect the points where oxygen was evolved. This setup was left for some time. So, what will happen? Photosynthesis took place. Which of these will undertake photosynthesis? The cladophora undertook photosynthesis. And after some time it was noticed that you can identify red, orange, yellow, green, blue, indigo and violet colors. It was found that in the blue region, I am against the blue, correctly against the blue. See, this is the blue region. See, I have traced it in continuation with the prism. In the blue region as well as in the red region, see, in continuation with that spectrum, see, the blue and red. I will just simply highlight the other green. Green is here, then just above green there is yellow, and just above yellow is orange, and below the blue is indigo, and below indigo is violet. Okay. So now what he noticed here is the aerobic bacteria was highly concentrated. They got accumulated in the blue regions and in the red regions. So if there were say thousands of these bacteria, they got accumulated here and here. From the other regions, they left the other regions. See, the other regions, they were equally distributed. I am removing it from the other region, the black spots, which indicates the bacteria were not present in the other regions. Okay, they got, all of them got accumulated in the red region and in the blue region. It's not that they multiplied, they got accumulated. Why? Because oxygen was evolved in these regions. Oxygen was high in these regions. Why is oxygen high there? Because the cladophora present in the red region, the rate of photosynthesis was high there. Okay, therefore a lot of oxygen was evolved, whereas here there was a cladophora, the rate of photosynthesis was low. So rate of photosynthesis is very low. I am indicating with low with arrow down in the other regions of the spectrum, but at red region and in the blue region, the rate of photosynthesis was high. And you know, when the rate of photosynthesis is high at that region, what happens? Evolution of oxygen. Oxygen is a byproduct of photosynthesis. Therefore, a lot of oxygen was liberated in that region. And because there was increased availability of oxygen in that region, what happened? The aerobic bacteria all got accumulated in those regions. From different areas, it came to these regions and became concentrated only in the red and blue region. So we could see a lot of aerobic bacteria were in the red region and in the blue region. From the other regions and all, which way they were equally distributed, they moved off and got accumulated. So from this, what we infer is that in the red and blue region, photosynthesis is high. This experiment proves that in the red region and in the blue region of the spectrum, the light spectrum, the rate of photosynthesis is high. Definitely, even in other regions, the photosynthesis is occurring, but a very low rate. This was proved with the help of this experiment. Once again, with the help of a prism, light was split into its 7 colors and was made to illuminate a suspension of cladophora and aerobic bacteria, and was left for some time. The cladophora undertook photosynthesis. The cladophora in the red region, which were present in the red light region, had the advantage. The cladophora here in the red region and the cladophora in the blue region had the advantage. The rate of photosynthesis was high, and therefore when the rate of photosynthesis was high, a lot of oxygen were present in the red and blue region, and therefore our aerobic bacteria, what happened? They all got accumulated in the red and blue regions. So from this we can prove that photosynthesis is high in the red and blue region of the light spectrum. This was proposed by Angelman. Last is not an experiment, it is from experiments we are deriving at a hypothesis or conclusion about which pigment is the chief pigment. We know in photosynthesis pigments are involved, and what are pigments? Pigments are molecules that absorbs light at a specific wavelength. There are 4 main pigments. They are chlorophyll A, chlorophyll B, xanthophylls and carotenoids. So the 4 pigments in plants are chlorophyll A, which is a bluish green pigment. Chlorophyll B is a yellowish green pigment. So both are green, only a slight difference in the shades. Xanthophylls is a yellow pigment and carotenoids are orange pigments. So the specialty of pigments is that they absorb light at a specific wavelength. Clear? Now, from different experiments, the absorption spectrum of each and every pigment was obtained. Absorption spectrum of A, absorption spectrum of chlorophyll B, absorption spectrum of xanthophylls, and absorption spectrum of carotenoids were obtained, and they plotted in a graph. So we can see the graph. So this is a graph. See, this is a graph depicting the absorption spectrum. So absorption spectrum of the different chlorophylls. Chlorophyll A is indicated by the green color. So I will use the same green and just see the graph. So chlorophyll A, see, it shows its peak where at 400, and then it goes down, its absorption goes down, and again it peaks up at 700. So chlorophyll A, you can see its peak is at nearly 400, close to 400, and at 700. Then chlorophyll B, the action spectrum of chlorophyll B with yellowish green color, see, it peaks up, see, where near to 500, say maybe 470, then it comes down and again it peaks up before 700, say at 670. Then carotenoids, which is shown in yellow color, see, the peak is at 500, look, its peak is at 500, and again there is no second peak. So like that way, peaks means graphs of the different chlorophylls were obtained, of the different pigments were obtained and plotted at the same time. So the first step was graphs plotted for each pigment. Pigment A, B, xanthophylls and carotenoids, and it was plotted against the wavelength. Wavelength is taken on the x-axis, and on the y-axis the percentage of absorption. Now the next step is the graph of the rate of photosynthesis. Rate of photosynthesis was plotted. Again, the rate of photosynthesis on the y-axis, it indicated the percentage of absorption, and on the x-axis same way the wavelength. Same way, but this is the case of photosynthesis. The rate of photosynthesis was plotted, and it was found the rate of photosynthesis was very high at around 400 and also was high at 700. So approximately 450 and 700, the rate of photosynthesis was very high, the peaks were obtained. Okay, so now we have these two graphs. Now the third step was very crucial. The third step deals with superimposition, that is, each graph was made to be superimposed on this graph. So this is the graph, photosynthesis graph. I am taking the photosynthesis graph. On this graph, each of the graph of chlorophyll A, graph of chlorophyll B, the absorption graph of carotenoids and xanthophylls was superimposed. Okay, and it was found when they superimposed the graph of chlorophyll A. So I am going to superimpose this. This action means the process. What I am showing, I am superimposing it. See, this is the graph of chlorophyll A. See, I am just jerking this for you to focus on this graph, which is of chlorophyll A, action spectrum of chlorophyll A. This is going to be superimposed on the absorption spectrum of photosynthesis. See, I am superimposing, and I must see that the x and y coincides, then means the graph is correct. It coincided, and now you look. you can see correctly it coincides. You can see the peak of chlorophyll A correctly coincide with the peak of the rate of photosynthesis at 450. At the same time at 700 also you can see for both photosynthesis and the chlorophyll A, it correctly coincides. So, from this what can we infer? Once again I am repeating, I superimposed. When I superimpose, must see that correctly it is aligned. X and Y axis should be correctly overlapping, and see the graphs. You can see the blue color which indicates chlorophyll A correctly coincides with the rate of photosynthesis, which is indicated with red color. Rate of photosynthesis peaks up at 450 and peaks up at 700. Similarly, the absorption spectrum of chlorophyll A, you can see the blue also peaks up at 450 and peaks up at 700. From this we infer that chlorophyll A is a major pigment. Chlorophyll A is a major pigment, meaning it's the pigment that absorbs the light rays during photosynthesis. Of all the pigments, where was it coincided with A, which means chlorophyll A is a major pigment. Chlorophyll B had its peak at other wavelengths, xanthophyll had peaks at other wavelengths, and carotenoids at other wavelengths, not correctly corresponding or coinciding with the rate of photosynthesis. From this we can infer that chlorophyll A is a chief pigment. Meaning of chief pigment, it is the main pigment that traps the sun's light during photosynthesis. So, and we can see there is only one chlorophyll A which is surrounded by the other pigments. Now, what I am drawing is which pigment? It is the xanthophylls, which is yellow in color. Then I am drawing the carotenoids, which is orange in color. And then I will draw the yellowish green pigment, that is the chlorophyll B. So, by way of color I showed you a complex. This complex is called the light harvesting complex. It comprises hundreds of chlorophyll B, xanthophylls and carotenoids, but only one chlorophyll A, which is bluish green. Only one chlorophyll A, which is the chief pigment. Chlorophyll A, I have to give a tinge of blue. Okay, I don't have the apt color, so chlorophyll A, there is only a single pigment at the center, that is chlorophyll A, which is the chief pigment which absorbs light, which is a major pigment. Then what is the role of the other pigments? We are saying this is the chief, this is the main pigment which absorbs light. What is the role of the other pigment? Yes, the other pigments also absorb light at various wavelengths. We saw from the graph, they absorb light at different wavelengths. So, the advantage is that all these light waves of various wavelengths are transferred to chlorophyll A, and chlorophyll A conducts or traps all the energy of various wavelengths. Now, we have a wider range of wavelength of incident light because of this, because the other pigments absorb light at various wavelengths, one at 600, one at 400, one at 300, one at 800, one at 750, one at 550. So, different wavelengths. So, we have a wider range of wavelengths. So, the other pigment absorb light at different wavelengths, and all these light energy is directed or transferred to chlorophyll A, and therefore, it makes the process of photosynthesis very efficient. Not only a wavelength which is coinciding to chlorophyll A, which is at the red and blue regions, at the other regions also all the wavelengths are directed to chlorophyll A, and all the energy is passed to chlorophyll A, which is the chief pigment and it's a single pigment. So, it makes the process of photosynthesis very efficient because we get a wider range of wavelengths energy directed to chlorophyll A. Another important property of the other pigments in the vicinity is it protects chlorophyll A from a process called photooxidation. Because of the other pigments in the vicinity, it is protected from destruction by the sun's light. Sun's light will cause photooxidation, will destroy this chlorophyll A, but because of the other pigments like soldiers, it is being protected from photooxidation. Clear? So, with today's with this video, you are clear on the experiments, pioneering experiments towards photosynthesis. That is, oxygen is evolved during photosynthesis, light is necessary for photosynthesis, then photosynthesis occurs in the red and blue regions of the visible light, and lastly the chief pigment is chlorophyll A. In 10th standard you all have learned that carbon dioxide is necessary for photosynthesis, light is necessary for photosynthesis, and chlorophyll is necessary for photosynthesis. But for 11th standard, is this part of the equation that is necessary to be studied. So, I hope you have understood today's experiment in the video. Thank you.